The gut is no longer seen merely as a digestive organ. It's increasingly recognized as a sensory, cognitive, and behavioral modulator, shaping not only hunger and satiety, but aspects of human psychology previously assumed to be housed exclusively in the brain. As the gut-brain axis comes into sharper scientific focus, one of the most provocative frontiers now emerging is the idea that gut microbes, via the neurobiotic pathway, can influence social behavior, risk-taking, and decision-making. This expansion of the neurobiotic sense suggests we may need to rethink how biology and behavior intersect, not as distinct realms but as deeply interwoven systems where microbial whispers help steer choices we think of as purely personal or psychological. At the core of this model is the bidirectional communication between the gastrointestinal tract and the brain, primarily mediated by the vagus nerve, gut immune cells, microbial metabolites, and specialized sensory cells like neuropods. These neuropods can detect microbial activity and chemical cues in the gut with stunning speed, passing information upward to brain regions involved in emotion, motivation, and social cognition, areas like the amygdala, insula, and prefrontal cortex. What's increasingly clear is that these neural pathways don't just regulate appetite, they help determine how safe we feel, how we assess risk, and how we engage with others. For example, multiple studies have shown that alterations in the gut microbiome can shift levels of key neurotransmitters like serotonin, dopamine, GABA, and oxytocin, all of which are intimately involved in trust, social bonding, aggression, and impulsivity. Gut bacteria can produce or stimulate the production of these compounds, and neuropods, as well as enteroendocrine cells, can sense them and relay signals to the brain. This is not a slow hormonal drift, it is a neural, real-time feedback loop. In rodents, specific bacterial strains like Lactobacillus rhamnosus have been shown to reduce anxiety-like behavior and increase exploratory drive, but only when the vagus nerve is intact. Cutting it breaks the effect. In humans, early trials with targeted probiotics have demonstrated changes in emotional reactivity and decision-making under stress, suggesting that microbial influence scales up into cognition. But the implications extend far beyond mood. Social behavior, how we interpret facial expressions, how we respond to group dynamics, how much we trust a stranger, may all be modulated, at least in part, by signals originating in the gut. The insula, which receives dense input from the vagus nerve, is a key hub for both interoception and empathy. If microbial patterns shift due to diet, antibiotics, stress, or inflammation, it could dampen or distort this input, subtly altering how we perceive ourselves and others. This may help explain why gut dysbiosis is frequently observed in conditions like autism spectrum disorder, social anxiety, and even antisocial behavior. Risk-taking is another domain ripe for neurobiotic influence. The prefrontal cortex, which weighs long-term consequences and short-term rewards, is deeply sensitive to both gut-derived dopamine precursors and vagal input. An inflamed gut, or one lacking microbial diversity, may amplify impulsive decision-making by heightening stress reactivity and reducing reward sensitivity. Conversely, a balanced gut microbiome may promote more measured risk evaluation, emotional regulation, and prosocial decision patterns. This could reshape how we understand not only individual behavior, but also broader social phenomena like economic choices, addiction vulnerability, or conflict resolution. Importantly, the gut's influence isn't deterministic. It doesn't erase free will. Rather, it biases the brain's default settings, shaping the tone of the cognitive environment in which decisions are made. A person with a healthy, well-signaling gut may be more likely to experience calm curiosity when entering a new situation, while someone with a disrupted microbiome may experience hypervigilance or avoidance. These subtle shifts, repeated daily, add up to long-term behavioral patterns that appear stable but are, in fact, biologically modifiable. This emerging science also reframes early development. In infancy, the gut microbiome co-develops with the nervous system. The microbes a baby acquires, through birth, feeding, environment, and antibiotic exposure, can influence not only immune priming and metabolism, but early social engagement and temperament. Animal studies show that germ-free rodents have altered social behavior and impaired fear conditioning. Reintroducing specific bacteria can rescue these traits, suggesting that microbial signals are foundational, not supplemental, to brain wiring. In the long run, this gut-brain social loop offers therapeutic promise. Neurobiotic interventions, from precision probiotics to vagal stimulation to diet microbiome optimization, could become tools not just for digestion or weight loss, but for behavioral health. Treatments for anxiety, compulsivity, or even impaired trust might one day include gut-focused strategies that restore not only microbial diversity, but the communication fidelity between colon and cortex. Social dysfunction may no longer be seen as purely psychiatric or psychological, but as something that starts in the visceral interior and works its way outward into emotion and interaction. In short, the gut doesn't just feed the body, it feeds the brain's social and emotional landscape. Microbial signals, carried by neural pathways, shape the backdrop against which every choice unfolds. As we learn to decode and harness this dialogue, we may uncover one of the most unexpected and powerful tools for improving mental, emotional, and relational health: the quiet but constant voice of the gut, influencing not just what we eat, but who we become in the world.